Thank you. Thank you. So, good morning. Good morning, ladies and gentlemen. My name is Delvin Wenani, and today I'm privileged to be here once more. And today I want to take you through the first, we are doing our first remote learning today for this term. And today I want to take you through I had started last term by looking at genetics. And today I'm privileged. I want to move forward with the same. And today I want to talk about genes. Genes. Immediately after the introduction, we are now moving to the genes. So those ones who are writing, we can write genes. So we are going to talk about genes in our 40 minutes lesson. And before we start our lesson, I want us to go through what we call the table of contents. What are we going to discuss in this 40 minutes lesson? So our table of contents look as I've displayed there. We shall look at the first thing, we shall look at the introduction. We shall look at the introduction to genes, and perhaps in the introduction, we shall also have the definition of the word, the gene. Then we shall also have the structure of the gene. That's our second content. We shall look at the structure of the gene. We shall also look at the DNA. We shall also look at the RNA. And we perhaps at the end, we shall also be looking at the differences between those two, the DNA and RNA. And we shall also look at the role of the gene, the role of the gene. So ladies and gentlemen, now let us proceed. Now, the first one, I had said we are going to look at the introduction, the definition of the word gene, the definition of the word gene. So let us move forward and look at that. So we're looking at genes. What are genes? When we were doing our introduction, I talked about chromosomes. And I said chromosomes, they are found inside the nucleus of a cell. Now, on those nucleuses, on those chromosomes, we have other factors we call the genes. And we're going to look at what now the genes are. Now, in simple, the genes are hereditary factors. We can call them hereditary factors. We can call them hereditary materials. And these ones, they are the ones which are responsible in transmission of they are responsible for transmission of characteristics from parents to the offspring. They're the one who transmit characteristics from the parents to the offspring. Now, they occupy a definite position on a chromosome. I had talked about a chromosome. So genes are found on a chromosome, and they are the one which will now carry the structure and maybe the features and the characteristics of the parent and transmit those features and characteristics to the offspring. And this is because of what we call sexual reproduction. Remember, the genes transmit the characteristics only through sexual reproduction. That's where the male and the female gametes fuse. Now, next, I want to talk about the structure of this, the structure of a gene. So how does a structure of a gene look like? If I can give you something and maybe ask you, what is that? Are you able to locate and say, this is a gene from the structure? Now, a gene, in the terms of its structure, the structure of a gene composed of the following. Number one, they have what we call a 5-carbon sugar. They have a 5-carbon sugar. They have a phosphate molecule. They have a phosphate molecule, and then they have a nitrogenous base. We call them the organic bases. Now, if anything I give you and it has all those ones, it has a 5-carbon sugar, it has a phosphate molecule, it has a nitrogenous base, in organic bases, then that one qualifies and we call it a gene. We call it a gene. If you go back to your book, they have elaborated most on that one, and it's deeply explained. It's deeply explained on how each of those one, like the 5 carbon sugar. One, two. I'm sorry for that. The 5 carbon sugars. We also have the phosphate molecule, and also we have the nitrogenous bases. They have already been explained deeply in your books. Now, we shall now go deeper. I want us to go deeper and look at the nitrogenous bases. The nitrogenous bases in deep. Now, the nitrogenous bases, these are made up of the 4 bases. And because when you look at the structure of, I'm going to display the structure of the gene, and you'll be looking at it, structures I'm talking about. You'll see those structures I'm talking about. Now, the organic bases are as follows. Number 1, we shall be looking at something known as the adenine. Adenine is abbreviated with the capital A, that is adenine. Then another base, organic base, is thymine, thymine with capital T. So adenine, capital A, thymine, capital T. And then another nitrogenous base is cytosine, cytosine, capital C. And then we have another one known as guanine. Guanine is capital G. So these are the organic bases, and this one are the one which composed of DNA. They form the gene. They form the gene together with what I had talked about, together with the 5 carbon sugars, with the phosphate molecule, with these organic bases. Now they form the gene. They form the structure of the gene. Now, very fast, I want us to look at now the 3 components. These 3 components I've talked about, they form what we call a nucleotide. And the nucleotides form the basic structure units of a nucleic acid. For example, the DNA. You have heard of the DNA. And this is from the 3 components that form the nucleotide. Now, the DNA molecule is made up of several nucleotides, which are formed together to form chains known as DNA strands. Now, I'm breaking it down. The DNA molecule is made up of several nucleotides. Now, I've said the 3 components that we have talked about, they form a nucleotide. Now, the nucleotide will now form the basic structure of a nucleic acid known as a DNA. Now, when you take several of the nucleotides, then you get a DNA molecule. So DNA molecule is coming from the 3 components of the nucleotide, which are coming together to form the DNA structure. And then those nucleotides, they join to form a chain, a very long chain, and we call it a DNA strand. We call it a DNA strand. So I'm introducing another term, that is a DNA. So what is a DNA? Now, I'll continue first to say the strands occur parallel to each other, and they form what we call a twisted ladder. They form something like a twisted ladder, which is a double helix. Double helix. If you check on my slides, just be keen with my slides, you will see on the left, you see I've tried to put a photograph of a structure of a DNA. You can see the way they are twisted. They are forming that twisted, and they form what we call a double helix. There's the main one here. There's another one here. So they are twisted together and they're forming. These are 2 strands. Those 2 strands twisted. You can see the way they are twisted, like they're forming something look like letter S. Very much twisted. Now, I was saying, and let me continue, that in the DNA molecule, now, I'm talking about the DNA. I've talked about the 4. I've talked about the 4 organic bases, but in the DNA molecules, the 4 bases occur in pairs. Now, in the DNA molecule, the 4 bases occur in pairs. What are those pairs? Number one, the adenine will always pair with a thymine. Adenine will always pair with thymine. Then the cytosine will always pair with guanine. I repeat again. In the DNA molecule, I've talked about the structure of a DNA. I've talked about the structure of a gene. But when we are going down into the DNA, you find that because of the nitrogenous bases I've talked about the 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. Then because now they have to form a twisted ladder, they have to pair up. So one should be on one side and then the other one should be on another side, the bases. So adenine will always be pairing with thymine. And then cytosine will always be pairing with the guanine. And therefore, as that one continues, then they form that twisted ladder downwards. A good example is what I've displayed there. That when we have an A, then it pair with a T. Then you have a G, will pair with a C and vice versa. Then T pair with A and vice versa. And C will also pair with G and vice versa. So whatever the case, you can start with G pairing with a C, then C pairing with a G, or in the other case. So that one, that is a pairing in the DNA. Then this is an example of what I'm talking about. You can see, and what holds them, what holds these two bases is a very weak hydrogen bonds. They have bonds in between them. And those bonds are the one which holds the two organic bases together. So that one is on this side and then the other one is inside. Then they being held by a hydrogen bond. You can see like now what we have here, we have a G, which is a guanine. Then we have a hydrogen bond in between here. Now this hydrogen bond is very important. I'm insisting on the hydrogen bond because as we move forward, I'll be talking about how the hydrogen bond is broken down so that we can untwist the DNA. So this is a hydrogen bond. Then we have a guanine, then pairing with a cytosine, a thymine pairing with an adenine. Then you have a cytosine pairing with a guanine. Then you have an adenine pairing with a thymine. Okay, they're around what we call the base pair. Then we move on. There's also another nucleic acid. There's another nucleic acid called ribonucleic acid, or what we call the RNA. Now, the RNA, in RNA, mostly if you want to know the differences between now the RNA, which is a ribonucleic acid, and then the DNA, you look at the organic base, thymine. Thymine is in DNA, but the thymine will be replaced with uracil. So you'll have another organic base, uracil. Whenever you see a strand, any strand, but when you look at their bases, the base structure you find, it has a U. It doesn't have a T. It has a U. Then it tells you that that one is an RNA. It's not a DNA. It's an RNA. And I'll be explaining it deeper, the function of the RNA, when I'll be talking about the protein synthesis, the function of one of the functions of the DNA, which is protein synthesis. I'll be talking about that replacement of thymine with uracil. So that is the structure. That is the structure, how it looks like. It has some codon. I'm going to talk about the codons. There are some codons. Codons occur in pairs. There are 1, 2, 3, 1, 2, 3, 1, 2, 3, like that. So those are what we call the codons. Maybe I can just talk about it. The RNA here, the RNA, majorly, we call it, when you go to your textbook, it's called the messenger RNA. Now, the function of RNA is actually to work between. Remember, when I was starting, I said, and I want to take it back a little bit, the DNA is found in the nucleus. DNA is found in the nucleus. But synthesis of protein is done in the cytoplasm. I repeat again. DNA is found in the nucleus, but synthesis of protein is done in the cytoplasm. So cytoplasm is outside the nucleus. So now we require a communication medium between, because now the function of DNA is to it's to help in doing what? In the protein synthesis. They say that it translates the genetic information into characteristic of an organism through protein synthesis. So for protein synthesis to take place, we require this RNA so that the DNA can be able to command protein synthesis to take place in the cytoplasm. So the RNA will now carry the information from the DNA inside the nucleus and transmit it to the proteins which are found in the cytoplasm. And that's why we say the RNA is a messenger. When you check the books, you see it's written with a small m, messenger RNA, messenger RNA. Now, how does it carry the information? How does this messenger RNA carry or transmit the information? So this is what happened. Once the DNA wants to communicate to the proteins in the cytoplasm, then what it does, it employs the RNA. So the RNA will come in and it will behave as if there's a replication, that's a duplication. So in this case, the DNA strand will form an RNA on the side. Now, the RNA I've said will have the same structure as the DNA, will have the same structure as the DNA, except where we have thymine, it shall be replaced with uracil. And in this process, the RNA will be able to copy itself, will copy the way it will replicate exactly the way the DNA is. But now it will be replacing thymine with uracil. So at the end, what we shall have a strand. We shall have a strand of an RNA, which has all the best organic bases of the DNA in the nucleus, except that the thymine in this case has been replaced by uracil. Then that information will now leave the nucleus. The information will leave the nucleus, now move to the cytoplasm. Now, upon reaching the cytoplasm, remember this information is in what we call codon. It's in what we call codon. That is, it's a set of base which is written in a triplet known as codon. So codon will now have, because now codon will be understood by a particular amino acid of the protein molecule. So that if it is a protein which is supposed to function on a structural protein, it will go to a structural protein. And if it's a code that has been sent to functional protein, it will go to functional protein. Because remember, DNA is helping in protein synthesis. But in protein synthesis, we have two types of proteins. We have structural proteins, which now determine the structure. And then we have what we call the functional proteins, such as the enzymes and hormones. And these ones, they control the functioning of the cell. So this coding, we call it codon, which is now occurs in triplet, will now be able to be specific. Where are you going to synthesize? For example, we have examples. If you check, we have examples like codons like AAA. That is capital A, capital A, capital A. That one is for is a coding for amino acid known as what? Phenylalanine. Then we have triple T is for lysine. We have C, capital A is for yaline. And then you have CTA for aspartic acid. Okay. Now, I think I've explained much about that one. So once the information reaches, once information is translated, now the information from the RNA will now be translated by the ribosomes. Remember, there are the ribosomes that will translate the information from the RNA. RNA is carrying the information from the nucleus, from the DNA inside the nucleus. So the information on the messenger RNA will be translated by the ribosomes in the nucleus, and therefore it shall be used now to assemble the amino acid into specific protein molecules. That's why we talk about DNA. We talk about DNA functioning as a protein. It helps in the doing what? In protein synthesis. Now, the protein molecules will therefore determine the inherited characteristics in organisms. The protein molecules will therefore determine the inherited characteristics in organisms. Then next, now let me talk about the role of the DNA as a genetic material. What is the role of DNA as a genetic material? Number one, it stores genetic information in a coded form. It stores genetic information in a coded form. I want to repeat that one, and I'm using the word coded form. That means the information that is stored cannot be interpreted by any other thing. That's why we are using the word coded. Coded means it can only be interpreted by the target. For example, if the information is transmitted maybe to a specific it's only that protein that will be able to decode that information and change it into a characteristic. So that's why we say it is transmitted in coded form, so that something in between cannot be able to understand the information being transmitted. Number two, it enables transfer of genetic information, which is unchanged to the daughter cells through replication. I'm going to talk about replication on the next slide. Then it also translates the genetic information into characteristics of an organism through protein synthesis. Now, I've said the DNA stores information in genetic, information in a coded form. But again, the same DNA will now translate the genetic information into characteristic of an organism through protein synthesis. I've talked about so much about protein synthesis, and that is one of the roles of the DNA. Now, next, I want now to talk about DNA replication. After looking at the three major functions, I want us now to look at the role of the DNA replication. DNA replication. Now, what is DNA replication? DNA replication, I want to define, this is the process by which, and I go again, I take it again, this is the process by which the genomes, that's the DNA, is copied in cells. DNA replication is the process by which the genome's DNA is copied in cells. Now, this is what I want to take it through. Now, before a cell divides, it must first copy or replicate its entire genome so that each resulting daughter cells ends up with its own complete genome. Ends up with its own complete genome. And I want to do something here. I'll be able to explain the next diagram, and I'm going to explain deeper on what I'm talking about here. This is now the DNA replication in action. DNA replication in action. Now, look at this. I want to start from this part here. This is C. That is cytosine. And this is one, two. Sorry for that. This is a C. That's a cytosine. And this is a guanine. You can see the way I was saying, Whenever you have a C, we shall have a G. Cytosine always pairs with a guanine. We have a thymine always pairing with a thymine. We have a guanine always pairing with a cytosine as we proceed. Now, replication starts from here. This is where we are going to talk about replication. Now, you can see, I had already said, kwamba, before a cell divide, it must first copy or replicate the entire genome so that each resulting total cell will end up with its own complete genome. Now, look at this structure. This is I'm talking about, I said again, it's a double helix. So we have two strands. There's one strand here. This is the first strand. And then we have another strand down here. Now you can see the way they are replicating. The first strand, which has an A, is moving away. Then another one also moving downwards. Now, in between, there's a complementary structure being formed. Now, this structure has, as I said earlier, once it separates, the structure in between will also have to come up with the pairs as we talked about. So if the original structure from the first strand had an A here, it will form it will pair with another T. If it has a C, it will form another G. If it has a T, it will now form an A, and continuing like that. Then the other structure, remember they were together. Then the other structure also will separate and form another structure. And the same thing repeats. If it's a, it will form a C down here. If it's an A, it will form a T down here. If it is a C, it will form a G down here. So what are we saying? We are seeing a new structure coming out. There's a strand down here with the former strand up here. So these two, this one, they will separate and form an independent DNA structure. And then the one down here will also separate and form an independent structure. So in the process, we shall have two DNA structures, one up here and then another one down here. So we have two structures. I think I'm very clear on that. So we have two structures being formed here. And this helps, sorry, and this helps in the transmission of, it helps in the maybe making DNA to be available for each of the daughter strands. So that is why when you look at this strand here and looking at the other strand down here, discover that the information has been passed on so that it has replicated. And the procedure repeats itself. The first strand here, the first, we are calling it now a DNA, this one, DNA 1, and this one we can call it DNA 2. DNA 2. Now, the DNA 1 will form an independent DNA, and perhaps the process of replication may take place again. DNA 2 will also form an independent DNA strand, and the process shall proceed again. So that is how replication occurs. That is how replication occurs. And maybe if someone has a question, I'll be able to clarify more on that one as we proceed. Now, maybe I want to take it again, maybe for the second time, because I know sometimes some students have problems understanding this replication. I want to take it again and maybe be a little bit clear on this. This is the original strand, DNA replication. This is a DNA strand, the original strand. Now, I'm marking it a half. I repeat again. From here to here, there is no replication taking place. So this is the original structure. Now, the process of replication is starting from here. Now, what is happening here? What is happening here? One, I had talked about something. I had talked about the relationship between the two thymine, the two organic bases. I said in between these organic bases, let me use this one. In between these two organic bases, there is a hydrogen bond. There's a hydrogen bond in between them. Now, for it to replicate, this weak hydrogen bond will disintegrate. This weak hydrogen bond will disintegrate. And once it disintegrates, it will now enable the strand here to unzip. It will unzip. Here it is zipped. Now it will unzip. Now from here, where we're calling it the helices, it has unzipped. Why? Because the weak hydrogen bond has released it. So the unzipping now will allow each strand now to replicate, to form another strand. Now, in the process of forming another strand, I'm saying I'll call this I'll call this the upper strand as strand 1. Can call this one strand 1. And then there's another strand here. We are calling it strand 2. Now, this is the first strand. This is the second strand. They are unzipped. And now, after this unzipping, the DNA strand, that is the base sequence, will be copied out onto a new DNA structure using the parent DNA as the template. And therefore, the parent DNA molecule is said to have replicated itself. So this is the parent strand DNA, strand 1. So it will do what? It will form another one. So if it is a T here, it will now form another one, which is an A here. If it's a G, it will form another C. If it's an A, it will form another T. If it's a C, it will form another G. Now, when you look at this, now it changes from being a strand, and it has now double-stranded. Now we form it. It forms what? It forms a new DNA strand, which is now double helix. The same thing with the lower one, the strand 2. Now the strand 2 will also do the same. It has the original, they call it the parent, which is G. For example, here it's A. Then it will form another one which has a T. The C here will form another one which has a G. The T will form another one which has an A. And that process continues. So it will now form, this one will now change from being a strand and will be a double helix, and therefore it will form a DNA. And therefore we shall say the DNA has replicated. The DNA has replicated. So allow me now to proceed. So next now, I want us now to look at the differences. Now, the differences between the RNA and the DNA. The differences between the RNA and the DNA. Now, an RNA, let me start with the DNA. I'll be reading one difference from both sides. So a DNA is a double-stranded nucleic acid, but an RNA is a single-stranded. When I was explaining protein synthesis, the RNA is working as a messenger. So what happens? It copies the structure of a DNA. So once it copies the structure of a DNA, then it replaces the organic base thymine with the uracil. Then it will now leave the nucleus with that information from the DNA. Now go to the cytoplasm, where now it can take that information to ribosomes. Now the ribosomes in the cytoplasm will interpret that information and therefore synthesize to specific proteins. Number 2, it contains deoxyribonucleic acid, deoxyribonucleic acid. But the RNA contains ribose sugars. This one contains deoxyribose. RNA contains ribose sugar. From its abbreviations, DNA is deoxyribonucleic acid. RNA is ribonucleic acid. So the DNA has deoxyribose sugars. Deoxyribose sugars, while the RNA has ribose sugars. That's the difference. The DNA has thymine as one of its nitrogenous bases. Thymine. But I said, when we look at the RNA structure, the thymine has been replaced with uracil. The thymine has been replaced with uracil. Another difference, it is in the presence of the nucleus of the cell. This is the DNA. It is present in the nucleus of the cells. And I've explained that one. But the RNA is present both in the nucleus and the cytoplasm. That's why I was saying most of the time we call this RNA, if I can write here, it is mRNA. This M stands for messenger. Messenger. That means it sends the information. Remember, it's in the DNA, it is in the nucleus, at the same time, it is in the cytoplasm. Now, protein synthesis is supposed to take place in the cytoplasm. So what the function of this does is to carry the information from the DNA, what the DNA wants, and transmit to the cytoplasm. So that's why it's both in the cytoplasm of a cell and also inside the nucleus of a cell. So ladies and gentlemen, I think with that, allow me to come to the end of my lesson today. And I cannot leave without giving some questions. And here are some of the questions that I want you to note down. Here are some of the questions that I want you to note down. Question number 1. I don't know why the question are not displaying, but I can be able to read to you. Question number 1 says, what is the basic unit of a DNA? What is the basic unit of a DNA? Then number 2, the first question was, what is the basic unit of a DNA? Number 2 is, name the chemical components of the unit you have named above. Name the chemical component of the unit you have named above. So the first question was, what is the basic unit of a DNA? And the second question is, name the chemical component of the unit you have named above. So ladies and gentlemen, thank you very much for your time. That one brings us to the end of our first lesson. Be blessed and have a nice day. Bye-bye. Or maybe if there's one question, maybe I can answer one, I can respond to one question, 1 or 2 questions because of time. Thank you. We can leave now.